Hello students, today we will discuss the light producing organs. Although luminescence is reported from a large number of insects, true or self-bioluminescence occurs in only some species of insects belonging to the four orders. Order Columbola, the insects of which are commonly known as springtails, Hemiptera and Homoptera, lanternflies, Diptera larva, especially the glowworms, and Coleoptera, that is fireflies, or Lampyris luceola. The luminescence may occur in both sexes or it may be restricted to female and even up to the immature forms, particularly in Lampyridae. In insects, the light is produced by the special light producing organs. The light producing organs are situated on the ventral side of the body and are developed from the fat body. They, however, differ in their number and location. As in the case of Columbola, the light is produced from whole body and the hypodermal fat layer functions as the photogenic organ. In case of the Coleoptera, mostly Lampyridae, the organs show wide diversity amongst the male, female and larvae. In the male of Photuris, there are two pairs of photogenic organs situated ventrally on the 6th and 7th abdominal segments, while the female possesses only a single pair of organs on the 7th segment. The larvae moreover bear no doubt a single pair of organs, but they are located on the 8th segment. In case of the Diptera, the luminous organs of Boletophila are exceptionally enlarged tips of all the four Malpighian tubules, which are lying in terminal segment of the abdomen. While in others, they are derived from the ventral hypodermal fat layer. In case of the Platyura, they bear a pair of photogenic organs at the caudal end, while the whole body of the larva and pupa of Ceroplatus is luminous. A large number of organs are reported in some of Coleoptera. There are three organs, a pair of lateral side of pronotum and a large median at middle of the first abdominal segment in case of the Elatridae. The Diplocladon larva possesses three photogenic organs, a pair of lateral side of pronotum and a large median at middle of the first abdominal segment in case of Elatridae. The Diplocladon larva possesses three photogenic organs, two lateral and one median on each abdominal segment, while the starworms bear a single at the center of each abdominal segment. In Phrixothrix, which is commonly known as railroad worm, lateral marginal photogenic organs are located on the second thoracic to the 9th abdominal segment, which emit the blue light. Besides these 9 pairs, two additional photogenic organs which emit red light, they are situated on the head. If we talk about the structure of the light producing organs, the light producing organ consists of photocytes, which are large size cells lying just beneath the epidermis. They form cylinders running at right angles to the cuticle. The dorsal cells form a layer over the photocytes. The cuticle overlying the light organ is transparent. The photocytes are richly supplied with trachea and nerves. Each trachea gives off branches at right angles which break up into a number of tracheoles running between the photocytes parallel to the cuticle. The trachea are placed at a 10 to 15 micrometer distance from each other, providing a short path for oxygen diffusion. The tracheal origin is enclosed within a large tracheal end cell. The inner membrane of the tracheal end cell binds the tracheoblast and is completely folded. The nerves entering the photocyte cylinder enter as a spatulate terminal process between the plasma membranes of the end cells and the tracheoblast within which the tracheole arose. The terminal processes possess two types of vesicle, large vesicles and small vesicles. The photocytes are packed with photocyte granules. The granules contain a cavity connected with the outside cytoplasm via a neck. These granules contain photogenic substrate, that is luciferin. Small granules occur dorsally and ventrally. Mitochondria are sparsely distributed in the photocytes. The dorsal layer cells contain urate granules and the cells form a reflecting layer. They also store the oxyluciferin irreversibly produced in light production. In photorests, two photogenic organs together contain about 1500 photocytes forming some 600 cylinders, each with 80 to 100 end cells. Then about the mechanism of the light production. Light is produced by the oxidation of luciferin in the presence of the enzyme luciferase. Luciferin is first activated by ATP in the presence of magnesium and luciferase to produce adenyluciferin. The adenyluciferin is oxidized by an organic peroxide, again in the presence of luciferase, to form so many excited adenyloxyluciferin, which decay spontaneously to low energy adenyloxyluciferin with the production of light. The energy for this process is obtained directly from the oxidation process, not from the ATP, and it is released in one large step. The radiation is very efficient, about 98% of the energy involved being released as light. The low energy adenyloxyluciferin produced inhibits further reaction by binding with luciferase. The pyrophosphate, however, removes the inhibition. When the light organ is stimulated by a nerve, the acetylcholine reacts with ATP and coenzyme to yield the pyrophosphate. The pyrophosphate diffuses to the photocyte granules and stimulates the production of light by removing the inhibition of luciferase. One molecule of oxygen is consumed per luciferin molecule, producing one quantum of light. According to Lee-McElroy, the pH induces changes in the luciferase structure in such a way as to give two colors of the light. The color of light produced by various insects is different. Lempiris and Photinus, they produce yellow-green light. Bolitophylla produces blue-green light and Fulgora produces white light, whereas Phryxothrix produces green and red light. Then what is the mode of emission of the light? A mode of light emission. There are various modes of light emission. giving different glow patterns. First of all, the continuous flow or the continuous glow. Light is produced without interruption as a continuous glow of uniform intensity, as in the case of larviform females of phenogods. Then there is intermittent glow. The glow lasts only for a few seconds, as in the case of Photuris. The light appears and disappears in a second. Then pulsation glow. The light is emitted in pulses ranging 6 to 13 pulses per minute in Pyrocella and 16 to 110 minutes in Luceola. Then there is flash of glow. The flash consists of a burst of light which remains only for a short duration with greater intensity. The light intensity in a flash rises abruptly from 0 to maximum and declines suddenly back to 0. Duration of flash is of order of 0.1 to 0.2 second with only one peak of maximum intensity and assisted by multiple ranging peaks. The flashing pattern is specific to species. For example, the true fireflies such as Photinus, Photuris and Luceola, etc. Then how the bioluminescence is controlled? There are two principal theories of neural control have been proposed, the tracheal neurocontrol theory and the luciferase neurocontrol theory. It is advanced by Snell and supported by Alexander. It holds that nerves regulate the opening of a valve in the tracheal end cell, which controls the inflow of oxygen, a key factor known to involve in light production. Buck considers that neural influence might act at the enzyme level and all the reactants necessary for luminescence are released to react by neural activity. Luciferase neurocontrol theory. It is proposed that the neural activity causes an indirect release of luciferase which reacts with the necessary substrate, all of which are present in excess. The flash is terminating when the enzyme is again sequestered in an inactive complex. The light organ is a true neuroeffector organ. The brain initiates the flash. Brain and peripheral neural complexes have inhibitory effect, while abdominothoracic ventral nerve cord has excitation effect. The inhibitory substance may be adrenaline in adults and neurotransmitter a monophenol resembling synephrine in larvae. Then what is the significance of light production? In Lampreyrids, significance of light is mainly in the case of for the three reasons. First is to attract the opposite sex, second is to lure the prey, and third is for the defense. In case of Lampreyrids, the light is a mating device to attract the opposite sex. In the fireflies, where both males and females are luminous, a fairly complicated signal system has been developed. Each species of firefly has a characteristic flash to which the female of the species can recognize. Photinus spiralis males fly about 50 centimeters from the ground and emit a single short flash at regular intervals. The female climbs above the grass and flashes only in response to a flash of light which is produced by the male. The female responses the male, producing light within an area of 3 to 4 meter radius. The male then runs directly towards the female and glows again. The male and female both emit light repeatedly 5 to 10 times until the male reaches the female and mates with her. In some fireflies, only one sex, that is female, is luminous, broadcasting a species-specific signal to non-luminous male, as in the case of Lampiris. Flash pattern of various species of Photinus fireflies differs with variable intensities and responses from the females. In Elatridae, female emits bioluminescence and pheromones so that males can be attracted. The luminescence of Boletophila larvae serves as a lure, attracting insects on which they feed into networks of glutinous silk threads which they spin initially. Thank you.